Get to the Flyte for biotech specialists. So I want to introduce here Niels Bantilan. Niels is an ML engineer at Union, and I asked Niels to give us a 8-minute, 10-minute overview about Flyte for biotech, just frame the topic, and after that intro, we'll move on and have the panel conversation. So Niels, the stage is yours.
Cool, thanks, Martin. Hi, everyone. I'm Niels Bantilan. So I guess I just wanted to set the stage here. I used to be in biology. I was in an immunology lab as a research tech for 4 or 5 years, and before that, I did developmental biology, basically taking care of South American opossums, and then mice after that. So I have a little bit of experience in a lab doing wet lab research. The key thing I wanted to just lay out here today, and I think a lot of the panelists later will have a lot more to say about this than myself. I guess before this talk, I just did a little catching up in the field, see what's the latest. But the main question I want to ask here is, what are the main challenges in computational biology and also biotech more broadly in life sciences that the next generation of tools need to address to make progress in this field? And the tool, like science and engineering, have always gone hand in hand, right? So we needed the microscope in order to make a lot of discoveries about cells and cell theory, things like that. You need a telescope to see out into space. So, and then in biology, like fluorescence microscopy, flow cytometry, all these things to get at what the cell is doing at a more granular level. Now we have computers, now we have a lot of analytical tools. So what does that look like, and what are the things that these tools need to do for us to make progress? So this is just a cartoon of basically the scientific method, but you can look at this and kind of replace different boxes for things that are specific to biology and biotech. You formulate a hypothesis, you do some experiment, you collect data in vivo, in vitro, in silico. And in my experience, most labs will have a bunch of multimodal data, like images, texts, like unstructured, semi-structured data. And it's all sitting in a computer somewhere. And, the researchers analyze it. And, it's all kind of local. There's not much way of, I guess maybe platforms now exist where you can do this, but you analyze the data and you falsify your hypothesis, you refine it, and then that turns eventually into theory and application. Oh, wow, there's an animation. I didn't do that. Okay, that was just a Flyte logo for some reason. So the main challenge is, from my perspective, and I think maybe the panelists can revise this, add stuff to this, but a lot of it has to do with, science needs repeatability in experiments. How do we overcome the reproducibility crisis? That's something that's been well documented so far. And, the higher level you go into like psychology and social sciences, this becomes even worse. Reusable components, like how much effort is being duplicated across labs and research teams and groups. Scalable analysis, wet lab in silico experiments generate a ton of data. Where do you store it? How do you process it? Going back into the reusability piece, collaboration between labs across teams, across organizations is a challenge. Complexity management, how do you connect and compose all the different pieces into some scientific workflow? And then data governance, who owns the data, who's responsible for maintaining it, maintaining quality, how's it transported across organizations. So in the next slide, I just hint at various things that we know Flyte does that might help in certain of these elements. I'm not going to read through all of these, but some of them including containerization, versioning of code and data artifacts. The fact that you can collaborate across teams with these isolated modular packages. Obviously, Flyte Kit itself is an SDK for expressing complex workflows. And then data governance, which is, I think, that's a piece that I'm very much interested in, but Flyte has granular data access permissions and certain baked-in data quality and documentation features. But that's where I'll end, and hopefully we'll have a broader conversation around this.
Awesome, fantastic. Thank you, Niels. So, I was thinking the topic is pretty much about data and analysis workflows, reading through your and listening to what you have said about reusability and reproducibility, scalable workflows, data governance. I think we have a whole range of topics today to discuss. With that, we have quite a few speakers today as well. So we have, of course, the team from Union, Ketan, who is the co-founder and CEO of Union, and Niels, who just heard Niels about his background in biotech. I also would like to welcome Jeev from Freenome, Kenny from LatchBio, Calvin from Embark, Krishna, Infinome, I always stumble over this, Krishna, and Jason from Zymergen, and again, congrats, Jason, to the recent corporate events there. So with that, I would like to introduce the first speaker, Krishna. Krishna, if you could unmute yourself and introduce yourself and give people a brief background why you're here today.
Yeah, absolutely. Thanks again for hosting this meeting. So my name is Krishna Yerramsetty. I am a data scientist at a very small startup called Infinome Bio. We're located in Boulder, Colorado, and Burlingame, California. So what we do is we're trying to speed up the traditional synthetic biology process, if you will. So people, if you're familiar with the DBTL cycle, design, build, test, learn cycle. So we're trying to speed that up, be more agile in some ways, and actually unlock some of the potential problems that we have right now in terms of different products that could be made using, let's say, E. coli or yeast. So that's the general motto of the company. So myself, I'm a data scientist. I'm not a software engineer. I went to school for chemical engineering, for of all things, and then I worked in biotech for close to 10 years now as a data scientist. So I've been working with Flyte a little bit for the past 4 to maybe 8 weeks. I don't remember exactly. And it's been super, super helpful for us because we are just a 2-person team. If you consider the CTO also as a data scientist / software engineer. So yeah, so Flyte and Kubernetes have been super helpful for us to get our NGS workflows up and running on the cloud, on AWS predominantly. So yes, that's been my experience. Is there more you want to?
Yeah, absolutely. I think it's fantastic. I mean, also, I think it's really amazing to have data scientists, not just infra people, but also data science people here in the panel today. So from what Niels said earlier about reproducibility, reusable code, and I think most importantly, data governance as well. Which one of those topics that you heard earlier are really on top of your list?
Yeah, I think reproducibility for us more than anything else. Data governance, probably if we are a pharma company, right? Like if we do a lot of patient data handling, for us, data governance is, I wouldn't say it's not an issue, but reproducibility is probably a bigger issue for us because we have so many different tools that we use, right? And each of those has different versions, and we need to make sure that everything is being used correctly, and the results that we have, let's say if we go back to those results a month later or maybe a year later, we know exactly what tools were used to generate those results and what code was used, obviously, to generate those results. So that's a higher priority more than anything else.
That's interesting. Great. Thank you so much, Krishna. So Niels, we just heard you earlier, but I think in this segment, please introduce yourself real quick as well, shortly. And then, I would love to hear from you a little bit about the specifics of Flyte, specifically when it comes to reproducibility, just like what we heard from Krishna about what you have discovered and why Flyte is a solution for that problem.
Yeah, so yeah, I'll reintroduce myself. I'm Niels. I have a background in public health as well, in biology, so I've sort of at the different layers of the stack of complexity of going from if you think of physics as the lowest level and then all the way up to society, economics, things like that. Yeah, it's reproducibility is a big problem. So especially in things like in public health, but even here where you're working with data and it's a little the problem with working with humans is always by observing them, you change their behavior. So it's like adds more complexity there. But even when you're studying cells and those lower level systems, like I think one of the big things that Flyte offers is, again, you're basically decomposing all the dependencies, resources, compute at the most granular level. And so you're never gonna foot gun yourself into making assumptions that, about your entire pipeline, and then one piece changes here at the end or the beginning, and then somehow your entire thing breaks. So that's one thing that I've come to really appreciate about Flyte, is that you can Dockerize at the node level. Isolate your dependencies. You can declaratively say in the SDK what compute you need, what resources you need. And I guess the one thing I'm super excited about is thinking about how you can do that with data as well. So I don't want to ramble on, but those are my thoughts.
No, I think that's really great insights, and we're going to get back to that in a little bit about the data analysis part of using Flyte for that. But next up is Jeev. Jeev, please unmute yourself and introduce yourself and tell us a little bit about how did you come to discover Flyte, and about your company and about yourself.
Yeah, absolutely. Thanks, Martin, for inviting me. Happy to be here and talk about Freenome and Flyte. So my name is Jeev. I work at a company called Freenome. We are a very cross-functional team that's essentially building next generation diagnostics for early cancer detection. And detecting cancer early is important because that's when it can really have an impact. We have better options for treatment at that point. And as a result, better outcomes for patients as well. But early cancer detection is a hard problem because, at that early stage, detecting cancer signal is really, really difficult. There's very little cancer signal respective to normal signal. And so we've built a multi-omics platform that assays a lot of signals, including tumor, non-tumor, and different molecular components like proteins and DNAs, hence the multi-omics, to try and get a better sense of the cancer, to possibly even before a tumor can develop. Right? And then we do this all based on a simple blood draw. So basically walk into your doctor's office as part of an annual physical. You get your blood drawn, to test your cholesterol or whatnot, and a similar blood draw can be administered. Our test can be administered with a similar blood draw. So we want to make it as accessible so that the test can be effective, right? A good test has to be both good in terms of its performance and how it's actually detecting stuff, and also accessible to patients. So within Freenome, I work in the research platform team. This is a large part of the innovation engine at Freenome. And we operate, and we support the different components like the compute platform, data platform, and automation platforms. And our role is to essentially empower our users, and our users span multiple teams across Freenome, including molecular research, computational biology, machine learning, and so on and so forth. And we want to empower them to basically build better products faster. And I guess we're here today because Flyte is a very large part of our research platform. It's a very key component of it. And that's just one of the places in Freenome that we're using Flyte. There are also other places, including things like our ETL, as well as our production pipelines.
Great. So I heard you say in the past, use the term production grade compute. What do you mean by production grade compute?
Yeah, I mean, so I believe that there are certain things that a compute platform should provide out of the box, that users shouldn't have to worry about, right? So from a user perspective, it should feel very simple, intuitive to use. So they just define their intent or their workflow in a very simple language like Python, right? Something that Flyte provides. And then they ship this workload off to a system that can be trusted to get this workload done and complete in some reasonable amount of time in a very resilient and robust way, and then give results back to the users. Right. And at the same time, if they wanted to ever go back and review that work and see what happened, look at logs, understand like how, the CPU and memory usages, were, with their workflows and stuff like that, they should be able to do that fairly quickly. And if, a year down the line, 2 years down the line, they wanted to just, go back and rerun that experiment to see if like there was any discrepancy, they should be able to just do that very easily. So these are all the things that I think a strong production grade compute platform should provide out of the box. And obviously, I'm glazing over like the obvious things like scalability and all that stuff, right? That is just like, I think, kind of assumed that that we need at this point for a compute platform.
Fantastic. So wonderful. So next is Kenny. Kenny, you're with LatchBio. So please unmute yourself and introduce yourself and tell us a little bit about the challenges that LatchBio is solving.
Hey, yeah, absolutely. Thanks for having me, guys. So I'm CTO at LatchBio. We are a SaaS platform. We service many biotechs at all different sizes, working with all sorts of modalities of biology. And we are the stopgap between where data is collected from the machine and where it's digested into human interpretable insights by the biologist. And so a unique challenge we face is like we're rolling out software to many types of companies. And so performance and scale is much less of an issue for the stage of companies that we work with, rather than reproducibility and the ability to deploy things on like heterogeneous compute environments with dependency management and so forth. Concretely, there's just a myriad of tools. Neil kind of alluded to this point that the bioinformatics tool is derived from a research process that is the person doing it isn't really trained in software engineering best practices. And so the tool itself is just going to be shoddily maintained on GitHub and hard to get up and running on a box on some cloud compute. So the process of like getting that containerized and rolled out onto a Kubernetes deployment, and then being able to click one button and have that go up again is invaluable in that name. So make this concrete, like we work with a lot of companies doing CRISPR discovery. And so a very useful workflow there is coming up with like a good guide RNA and for your CRISPR therapy to be able to like go after a gene of interest. That often it's a SIMD type application. You're scanning against the genome with the same sort of function, same sort of algorithm. You need a GPU to be performant. So to get that software up and running on a box with NVIDIA drivers, it takes a while. It's hard. And so Flyte lets us just roll out that Docker container to that instance on Kubernetes, and it works like a charm. So we've used Flyte as our core workflow orchestration engine. I've been working with the Union.ai team for almost a year now. And yeah, that's kind of the gist of it. I know I was kind of long-winded and scattered, but happy to dive into anything in particular.
No, absolutely great overview. Maybe one follow-up question: what kind of like workflows or loads, do you put in the system? I assume you're in the early stages of your company as well, but you also look forward and actually want to, have some scalability on the system itself.
Yeah, I mean, so like I mentioned earlier, the vast majority of biotechs are not really constrained by how fast you can process data, because their experiment turnaround times are far larger. The minimum between the two. The maximum between the two is always going to be the time in the wet lab. However, we do work, the largest loads come from genetic screens at the closest to clinical level. So companies doing whole genome panels against like multiple different candidates. And that sort of setup, you're going to have 1,000 to 10,000 whole genome assemblies. So each one of those assemblies is going to be a 100 gigabyte file. And then the actual operation is going to require a 96 core machine for each one of those files. So it's a lot of data moving, a lot of compute being spun up. And so we just leverage a node group on our Kubernetes deployment on which Flyte resides that has 50 odd 96 core machines, and rely on the Flyte system to schedule them on an as needed basis and reschedule if the spot instance gets preempted.
Wow, impressive. Thanks, Kenny. Next up is Jason from Zymergen. Jason, so please introduce yourself and tell us a little bit about your role and Zymergen.
Hey, Martin, thank you. So yeah, I'm Jason. I work at Zymergen. We focus really on strain engineering and strain improvement in order to develop new products. and biology is hard. So we try and do this through high throughput methods, run a lot of experiments, figure out what the biology is doing through that. And my role at Zymergen is I'm a solutions engineer. So that kind of means I wear a lot of different hats depending on what's going on, from more traditional software engineering to product management to support to tech training, all of the above. And one of the things I was involved in with this role is understanding our long-running jobs and like async execution architecture at Zymergen. Zymergen went through a period of really rapid growth. So we pretty much like doubled in size from like 400 to 1,000 employees like over a year. And kind of through that rapid expansion, a lot of different of our software development teams kind of siloed into different platforms for doing async execution and software development. After a while, this became a little unwieldy. Things became sort of less reusable, less scalable, harder to maintain. And so my job was figuring out, well, do we really need all this stuff? And, well, if not, what do we do about it? The result of kind of that effort was, no, we don't need all these different platforms. They're all kind of doing the same thing. And they all sort of had a core problem in that you had to deploy each system with all the workflows at once. And we're really trying to move towards sort of continuous deployment. And so that sort of made that a big challenge. And so when we get to well, what do we do about it? We sort of surveyed the landscape and found Union and Flyte, and really liked that each workflow was sort of the unit that could be versioned, deployed independently. If something breaks and we need to fix it, it's just that workflow we need to worry about. And it's also really easy to work with. So we've got a lot of different kinds of developers, traditional software engineers to scientists that have become software engineers to data scientists to bioinformaticists, scientists with a little sprinkling of Python here that want to blow up a CPU for a while. And so we wanted something that we could make that's really easy for them to use and sort of have all these different user groups working within the same system so we could get that sort of scalability and reusability. And so far, Flyte's been doing a great job at that.
Fantastic. Thank you so much. So Ketan, you're next. So I'm sure the crowd here knows Ketan is CEO of Union. And I think you wrote the first line of codes for Flyte back at Lyft. So please introduce yourself and tell us a little bit about Flyte, the genesis of Flyte, and now the adoption, the rapid adoption in biotech. Why do you think that is such a standout field and segment for Flyte?
Firstly, thank you. I'm not even close to all of you guys, like what you guys have achieved in biotech. I'm basically with software. That's what I've done. But it's been amazing to hear what all of you guys are doing, and it's like it's almost humbling. I'll tell you maybe a short story, like 2 lines. When I first started Flyte, I was solving my own problems really, right? And the problems were, I was actually handling pager duty for software, a model that a data scientist wrote that I had no clue about. And I was like, okay, I want to change certain parts of the model. How do I do that? That was one. Second part of the story was there was a data scientist who left my team, and I lost the model on his laptop. And I spent 3 months trying to build a model. And I had to go to the CEO of Lyft and constantly explain, oh, I'm working. Give me time. I'm just trying to rebuild the exact. And what we ended up doing was just rebuilding the exact same model for 3 months after spending a lot of money. And then the third one was this constant, none of the tools that existed really. But we actually had this fantastic model where we wanted to use 1 million parameters or something, right, for training some idea. And when the data scientist came up with the idea, we sat down and we're like, it's going to take 3 months for us to build that infrastructure. Can you wait? Let me provision machines. We have to get Spark clusters, and you need some other stuff. I have to write that code and what ended up happening after all these 3 is that we're like, why are we doing this consistently? Like I spent a year of my life doing nothing but kind of throwaway pieces of work and and really reduce the velocity of this company. So that was one. Secondly, my career, I've worked across a lot of different industries, and I ended up doing similar stuff. Even though, like in mapping, we were trying to build a map, we were like running on, this is 2012, I brought down S3. And one of the reasons, and that's how you learn more about the system, right? When you bring things down, you're like, oh yeah, there's a bunch of problems in S3. And the reason why we brought it down, because we hammered it so fast. And I found fun stuff in Spot where, like, if you kind of pin yourself to be 80% of an on-demand cost, you outbid everybody else from the market. This is 2012. Now they don't let you do this. And so I just said, okay, I'm going to pin at 80%. I like show 20% discount, and I'm going to spawn off 10,000 machines, and I'm going to create the world in like 30 minutes. And we wrote a bunch of software that orchestrated all of this stuff. And the point of the story is that we were just doing, like, many times I was just doing the same thing instead of solving my own problem. And it was the core genesis of why I didn't want to do infrastructure. So I had built cloud software and so on. When I joined Lyft, I said, I'm not going to do infrastructure. I just want to do this, like, mapping and ML stuff. But this problem kind of irked me again. And we're like, okay, let's try and see how we can solve it. Not going to be open source, just for Lyft. And I will be completely honest, I had no idea about biotech. I never worked with a biotech person, and never actually met a biotech engineer. And it's amazing to see that it's kind of helping solve these problems. And it's actually the right use of a tool like this. Like a tool or an infrastructure piece like this has to help not just make better recommendation engines and advertising engines, right? It needs to solve real world machine learning problems. That's what I think we are here for, and I'm like really happy to be part of. I heard a bunch of different folks talk about reproducibility, decomposition, declarativeness, SIMD, and it's amazing. When we were building, we talked about all of these. We actually made a bunch of trade-offs. We said, I like to use Kubernetes. This was a spectrum that was happening. It was like Kubernetes was just coming on 2018. Like, think about the Kubernetes was really early then. Lots of skeletons in the closet over there, and we were working with it, but we saw the potential. You could actually declaratively create infrastructure and connect pieces. And so we made some choices that meant lack of some flexibility, right? We couldn't do, and then we kind of engineered our way out of it by creating dynamic workflows. And so many of the things that you see today are basically things that we wrote in one document of 15 pages, but were not able to solve it over a period of time. And so I think it's great to see that they apply to biotech, but I have now talked, and I've been in an interesting position that I've talked with folks in different organizations, and they have all had the same sort of issues. And this is not just biotech, it's physics, biotech, machine learning, all kinds of different fields that kind of come together to solve a big problem. Like whenever there are complex problems to be solved, you need these sort of use cases or these sort of requirements from a system. So thank you for having me. Hopefully that helps.
Absolutely. So I think what we hear here today is like biotech is actually not just like one or two requirements. There's a complete set of requirements. And that usually warrants a platform approach to answer, because otherwise you end up with point solutions. And so my question for the panel right now is, how long did it take you to discover that the point solutions themselves are probably not good enough? You need to have an underlying fabric, a platform to really move forward in your journey of providing biotech services in your organization. So this is an open question, so feel free to jump in.
I can go ahead. My company Embark just recently went through a process of shifting a lot of our production data processing over to Flyte. And there we had a homegrown set of tools for our sort of like, we do direct consumer testing. So there's like the customer facing pieces of our computation that gets quite large with thousands of samples coming through a day. But there's another piece that's like research. And we found that we had our homegrown tooling that we were spending a lot of effort on for our sort of customer facing tools, but then we kept rebuilding things for research. And that was one of the things that really made us realize, like, oh, wow, this is both painful because when we want to translate stuff, it's a whole big ordeal, but also we're spending a lot of researcher time and folks who have hybrid skill sets. They're people who have, like, they went to school to do biology and they're tired of working on infrastructure, who are struggling through bash scripts to, like, we've had bash scripts that like spin up EC2 instances and try to do the clustered compute for someone's research work. So for us, that was a big part of it, was seeing that get like duplicated separately from production in what people were trying to build for research, and realizing like this doesn't make sense. That's going to harm that translational aspect of the research.
Fantastic. And so Calvin, you know, we didn't introduce you, but I wanted to get to you at the very last, because I think Embark has actually a couple of very interesting stories to tell. You and I shared a few things beforehand, but tell us a little bit about Embark and you know, how Embark discovered us, specifically with those, you know, fast, you know, changing requirements when it comes to, you know, having a platform in your organization. And it's not a small organization, I assume. It's Embark has been around for a little bit, right? So tell us a little bit about Embark, about your role and the needs for a platform orchestrator.
Sure. So Embark's been around for about 5 years, and we've grown pretty quickly in that time. We do direct-to-consumer pet DNA testing, so it's a little different. It's like 23andMe in the concept, but we focus on pets. We're also doing other things now besides just DNA testing. We're doing methylation testing and some other things. So we originally, like I said, had a homegrown platform, and around well, since the company started, we've been talking a lot about, like, is that the right solution for this? We looked at tools that existed like Airflow and some of the sort of like older orchestration tools, and we're never happy with, in particular, like managing complex science pipelines that stretch across teams. Like, here's the team that processes the raw data and extracts some more featureized version of it, and then here's the team that does the like machine learning piece off of that. Here's the team that predicts the breed of the dog or the ancestry of the person. Here's the team that finds relatives. Like, each of those were very different scientific skill sets, such that you couldn't collapse all those into a team. But managing these pipelines with the existing tooling was very hard. You really need like contracts between teams at that point that stretch beyond just like a JSON spec, right? Like, I've done this type of thing for e-commerce, and there it's like, okay, we have like a JSON object and we align on that. And here it was like, okay, we have like VCF files and these big binary files, and people need to understand the contents and what they mean in a much richer way. So we were doing a whole bunch of exploration about 6 months ago, and we were exploring Flyte, we were exploring Nextflow, we were exploring a bunch of these tools. And what really brought us over here was both the robustness of the like reference task structure, where you can have multiple teams deploying separately and then composing those things, but also the ability to build that contract and build integration tests that stretch between teams, and having robust like typed interfaces between those teams. I think that was part of what really got us interested and got us excited. And then we had all of those science problems that we also wanted to fit into the same framework, and it was one of the things that checked both our production boxes and our research boxes nicely.
I think that's a fantastic view to have, research and production, because I think in biotech we have to combine you know very different teams and skills. Research and data science and is one area, and then the production engineering part is a completely different one. So that's also an open question for the panel on getting teams together. If you look backwards through experience, what were kind of like the lessons? How did you get your data science team onto a platform like Flyte? Or if you come from the infra side, how did you actually, you know, get your infra team onto Flyte? I mean, and then you have to actually make sure that all of those teams together embrace the same platform. So looking backwards, what was the experience collaboratively getting everybody on the same page?
I can talk a little bit about that. So at Freenome, we have this mantra that, like, you know, we don't want to have a lot of tools in-house. Like, we want to have a few things that work really well, that we really understand, that we can, you know, we're very confident about operating, keeping up, debugging, fixing, and so on and so forth. And also, like, you know, in the case of Flyte, like we've built, I think, a pretty good relationship with the team, and like we feel very confident, like supporting this tool, like in our production stuff. I think the things to note because of this is that, like, we need a very flexible tool to like support both our research use cases and our production use cases. And that's essentially like one of the main things that we get from Flyte. And that allows us to like basically, go from research into production like fairly quickly. Otherwise, you're ending up like doing a lot of like rewriting or like modifying, large pieces of your code base as you're transitioning or promoting it from research into production. So all of those reasons aside, like I think that was like one of the like very important pieces for us to very important like reasons why we went with Flyte. I think like getting people onboarded. So we started, I think like around 2020 or so, after like Flyte had been open source, we were very early in our adoption. And like one of the first things that we did was basically like setting up essentially like a sandbox internally so that users can start playing around with the SDK and like really, have a very like isolated playground in which like they can just test things out, do regression testing even, right? Like check out specific features and so on and so forth. So we spun that up actually fairly quickly. And since then, we've had like our engineers were like, very like one of the first ones to like start adopting. Like we were at the point starting to write our production pipelines and stuff, or moving our production pipelines to Flyte really. And so like that happened fairly quickly. The research side moved a little bit slower. I think researchers are generally more resistant to change, like because they don't want to focus on like the UX and like all that stuff. They just want to like do their research in like an environment that is like most comfortable and familiar to them. And so, that took a little bit more work. But so the way that we started there was to start onboarding like specific teams and like working very closely with them to like, see them and to really empower them to be successful on this platform. And then as we started seeing like more success and people started getting excited about it, like there's a lot of word of mouth going around like within the research organization. And then other teams started picking that up, and then we started supporting those teams as well. So it was a little bit more, it was a little bit slower, albeit a little bit more like, it's like very mindful, intentional on the research side. On the engineering side, it was more of a no-brainer because of like all the great features that like Flyte provides us.
Fantastic, thanks, Jeev. So Krishna, you on the
so I actually want to jump in there really quick because part of the premise of what we're doing is like recognizing from a social engineering perspective. The way these companies work is you have folks with different levels of experience with computation and computers and programming. Like as Jeev described, I think Freenome's unique because they have such a high density of engineers, and that's really an outlier in biotech companies at large. There is this big chasm that exists between the people who work at the bench, who do the pipetting, or who are unable to like even run a Python script. And so one thing that we've done really with Flyte is we built a compiler to generate React code from the Flyte interface. So taking the typed interface in the Flyte IDL system, and we generate front-end interfaces for it so the web lab scientists can self-serve. That is, and then shipping a thin wrapper over the Flyte SDK allows people on the bioinformatics side to write their own pipelines, but then expose them directly to that research team. I'm actually incredibly curious how Freenome was able to expose the Python directly to a lot of people who don't have fluency in Python. Or maybe it's exposing the Flyte console directly to the biologists, but this is a huge problem in the space. And as you said, I think that's spot on. There's a huge inertia against adopting new tools because biology by nature is conservative because of the reproducibility crisis at the bench. So the people have, they're not exactly super eager to pick up new computational tools.
Jeev, do you want to respond?
Yeah, I'll respond briefly, and then we can also talk offline, Kenny. I think I would say, like, I do agree with your point. I think, like, just exposing those interfaces has been really key. So we do have teams that are responsible for building those pipelines and, like, essentially providing, like a configuration layer for our molecular researchers to, like, kick off these pipelines. And, it's not like, there's not like a React component to it yet, but we do have like UIs that users can basically log in to and like fill out certain configurations or like modify the configurations for the pipelines that they want to run, and then just kick off runs that way. So it is very similar to like what you were basically suggesting. Yeah.
Cool. Love to see some of that open source sometime.
So I would love to get Krishna back in here a little bit, and because Krishna, you're coming from the other side, from the data science side. So tell us a little bit about your experience for actually, getting onto board with the system now. Was it data science moving and research moving slower in your company as well, or were you actually kind of like an outlier moving faster than what we hear?
Yeah, so I think a couple of things I think with respect to that. So we, like I said, we are a small team, but we have tons of projects, synthetic biology projects going on at the same time. And one of the things is we want to self-serve, like Kenny was saying, directly to the bench scientists. And we don't want this to be the bottleneck, data science / software engineering to be the bottleneck between those iterative loops of DBTL, right? So learn is a key step in that whole loop: design, build, test, learn. And we don't want to be the slowest step, learn. I mean, we have tools now. We can talk about that in a different venue, where we've actually sped up the entire biology process much, much faster. So to a point, actually, where processing data is the bottleneck right now at some level. So we wanted to make that bottleneck go away. So having some kind of self-serving tools, like Kenny mentioned, directly to the bench scientists, exposing these interfaces and making sure that everything in the backend is reproducible and be able to scale to the number of samples is really crucial. So that is why I think, previously in previous companies, that was not a problem. Data scientists could take their time churning through the data, or bioinformaticians, whoever could take their time churning through the data, because wet lab was the slowest step. Right. So here in our company, the paradigm is a little shifted. So that's what prompted us to go beyond the traditional bioinformatics tools, where we were each building our own R scripts or Python scripts and Bash scripts or whatever, and trying to do the analysis individually. Each data scientist has its own environment / preferred tools. We want to take that away and make everything unified so that we can go faster. That was the motivation.
Interesting. So I want to get over to Niels. Niels, in your intro earlier, you talked a little bit about the conditions that are so specific and special in biotech. Now, listening to that conversation here right now, and then actually make it easier for the researchers to actually do their work and so on. So what was your experience so far engaging with biotech companies using Flyte?
Oh, sorry, what?
So the question is, listening to the collaborative aspect and also to understanding how to get the researchers on board and make work simpler and easier for them, and getting to results as an organization faster. So you're speaking to a lot of Flyte users out there. So I just wanted to ask you if there's anything that actually stands out where you can see a pattern there. We say, look, I hear this all the time, but this one is a very, very specific one that that is worth mentioning here in this context.
Yeah, the interesting thing is that I feel like there's nothing really, the things that I hear are at such an abstract, more computing level that it kind of applies to all fields, to be honest. So like biotech will have its own specific manifestations of these problems, but a lot of it comes down to, you have domain experts. They're not software engineers. They're not trained in, they haven't internalized concepts that we all take for granted, like versioning data. Like we had image files that were, named by date, something, and then final, and then underscore final, and then underscore, and then another date, so it's like the way scientists do their work is very different. And one thought I had here is I'm curious to see what the panel has seen in their own teams of kind of the hybrid people who are the translators between different disciplines and are able to sort of navigate the two different types of stakeholders, right? Like scientists versus software versus infra. And I feel like a lot of the stuff that can't be automated, our organizational change, adopting new tools, all these things, that's a piece that I'm very interested in learning more about. Because as someone who helps people debug their Flyte workflows and whatnot, that's not something I hear often, but I have a sense that that is a problem, based on this conversation as well. And very much interested in Kenny's thoughts here as well on what do low code, no code interfaces, and he's hinted at that already with the strongly typed workflows that you transpile over to different components using React or whatever other web framework will emerge. But I'm curious to see what that there's both a technical component of translating these things to more accessible interfaces, but then there's like the softer human component of how do you actually get, you know, I used to work at Memorial Sloan Kettering Cancer Center in New York, like we're very well-established cancer institute. Like, how do you get the scientists in those to adopt new tools like this?
Okay, is there anyone from the panel who wants to respond?
Yeah, just briefly, I think, you know, it is a huge problem. The lifeblood of biotech is academia. So to get the old guard to adopt tools, that has to be published in a paper. The best way to do that is to get in touch with people doing cutting edge bioinformatics and get them to adopt the framework that you are using. And you have to do that organically. So you have to build something that's incredibly useful and intuitive. I think that's a multi-year process, and it involves the same sort of user testing and on the boots interaction with these people that you would have in any sort of other field. Those principles haven't really been applied to biotech at large, which is why we haven't seen something like this. There's been previous attempts, tools like Galaxy, that have really floundered. But it succinctly, you know, academic biotech has to adopt these things, and to do that, they have to be confident enough to publish them. And that's how you get these older folks to use it.
Fantastic. So we have about 7 minutes left. I would like to ask one question to the entire panel here. Now you have, you know, hindsight is 2020. You have made your experiences. For folks who are just entering this, you know, the problem space and discovering, you know, taking the first steps, what is the advice, maybe the one line advice you want to give, you know, maybe a lesson you have learned that you want to share with, you know, other people in that space? Just going to hand it over to Calvin if there's a one-line advice from your side.
Sure. Don't be afraid to use the Slack community. It's a surprising one, perhaps, but we found a lot of times where we got really heads down in a problem when we're like reading the source code. And the Slack community has been incredibly robust for this, even though like it's outside of our domain or around like the particular problem we're working on. It's probably been one of the more robust, like this and DBT, I think, are like the two Slack communities that like, or like discourse in DBT's case, like there's very few things where it's like you can go ask a question, get answered in like an hour from like someone who's involved with the development team. So anyway, that's more than one line.
That's all good. And I know those people who answer the questions. I see them sitting next to me, and you know, Ketan and Niels obviously are some of those people as well. So that's fantastic. So Jason, from your point of view, the lesson that you learned and the advice you want to give.
Yeah, I'd say it seems really obvious, but don't reinvent the wheel. That's something we've learned the hard way over and over again. I think you get a lot of really smart people together and you're in that startup mode. And yes, we can all write our own async execution frameworks. We've done it before. Doesn't mean you should. It's worth, you know, even though you're trying to move real fast, to kind of explore what's out there, find something like Flyte that can meet your different use cases. To understand, you know, just the different user personas that are actually going to be using your system to not just solve your own problems, but your organization's problems.
Great, fantastic. Jeev, how about you?
Yeah, I mean, it's a hard question, but I'll say this: like, I think open source is like a big important thing in biotech, specifically because, like, you know, we are trying to build this like FDA regulated product, right? And so I think, like, for us, the more things that we have to build in house, you know, the more proof and evidence that we have to show the FDA that, like, you know, this is the right way to do things. I'd rather invest that effort in, you know, like a really solid, like, open source project like Flyte, and then, you know, grow that product, nurture that product for biotech, and, you know, for a future like FDA regulated products, together with a much larger like open source community. So like having one really nice standard that, you know, a regulatory body can just look at and be like, oh yeah, there's other companies in the space that are like using this and been successful using this, so it makes sense, right? It would be a much better like selling point than saying like, oh yeah, we decided to build this thing in-house ourselves. So like agreeing with what Calvin was saying about like community and the Slack community, and like, you know, nurturing this open source project, and also what Jason was saying about like don't reinvent the wheel. I think putting those two things together and then essentially working together as this group, right, like to, and future adopters of Flyte, to like make Flyte better and more usable for biotech applications.
Fantastic. No, I think that's also the main purpose here, having the community come together and actually establishing, you know, working on that standard. I feel like a great point that you made. Krishna, your advice for people entering that space?
It's tough to improve on what people have said. I really like what Calvin said. I think Slack is probably, something that really helped me moving so fast. So that's what I would reiterate, I think. And then just try it out. I guess, I mean, there's some problems. I think someone else mentioned that is sometimes, researchers are hard to, I mean, they have inertia. So you need to be really careful in how you actually talk about these tools, I think, if you are in a larger company with biologists. The things that I've seen, because problems with Nextflow and galaxies, I think there's a communication problem in terms of what these tools are capable of doing and not doing. So having that explicit conversation, both with data scientists, probably software engineers, and also biologists, is probably an important thing before you actually venture into adopting tools like this, making sure the contracts are explicit. Yeah.
Got it. And Kenny, obviously great insights from you and LatchBio. So what are the lessons, or maybe 2 lessons that you learned and advice you can share here?
Yeah, absolutely. I mean, if you're an engineer, get your hands dirty, get in the source code. Don't be afraid of peeking up behind the curtain, see exactly what's going on in all cases. I think too often we have a tendency to black box these tools. Let's not mistake ourselves. We're not the bioinformaticians here, we're infrastructure engineers, and it's our duty to really understand how these things work. So yeah, read a lot of the lines of the source code of Flyte. You guys did an amazing job at Union. Looking forward to keep on working and learning with you guys' team.
Fantastic. And Ketan, I'll hand it over to you to take those inspirations as well and the feedback, and it gives us a little bit of maybe a 1-minute overview about what's next here for Union and for Flyte, and how important the biotech is and will be.
Well, thank you, Martin, and thank you all for a wonderful discussion. I learned a lot, and I think at Union, at least on our side, we have seen a huge community being formed on the biotech side. Fantastic work by Kenny and Jeev and others who have actually helped kind of seed the community. And we would love to, we are here to help, but as I said, I have no real idea about all of the things in biotech. And what I would suggest is we should probably have a working group of some sort where people can, and to a point where Jeev made, and I think somebody else, Flyte is an extremely flexible tool, but it's just, and it was designed because of the flexible nature of science. And so we would love for, the tutorial section in the docs is maintained by folks within Union, and there are a couple other folks who have contributed to it. We would love that some of you actually add, bio with Flyte or a section like that, and then, help add, so that folks who are coming in can become really, on day one itself, can get use from it. We would highly appreciate that and recommend it. Another thing would be if there are specific features that I think we need within the Flyte core, or even on Flyte Kit and so on, that actually help biotech in any way. We would love to know that. It's just there's no way for anybody to know the problems unless you file the issues. Nobody feels bad about issues, and I've said this to multiple different people, because some people have said, can I really file an issue? There's issues are, that's how we are sharing problems and ideas, and even bugs are important to be filed. So please file issues, and I would love if we can, going forward, probably every quarter once, maybe have a small biotech section within the open source community thing, spend 15 minutes just talking about problems and what do you guys foresee, and what would you guys want to add to the core, and let's make Flyte for Bio one of the most important tools that exists, so that companies like you and more can actually get use from bio, get use from Flyte. And yeah, we are here to support. We are extremely community focused, if you have noticed that, and we would love to nurture and build this community. So thank you, and thank you for trying Flyte. The pioneers are the ones that actually build a community. And so thank you for all of you being the pioneers.
Fantastic. Thanks, Ketan. Thanks to our speakers here. This was very, very insightful, very great, very good conversation. So we're going to share this conversation online as well. I like the idea, Ketan, of having a work group, and I think we should pick this up in one of the open source things here as well. I think it's a fantastic idea. So with all of that, I just want to say thanks, have a great day, and we're going to publish the slide deck as well with the office hours and the upcoming event. So we'll see more information as soon as we get it posted. Thank you very much.